Welcome to Simplify Science Lecture 3.4, Search High and Low. We have a quote from Dr. Seuss underneath these stained cancer cells. The quote is, "I like nonsense. It wakes up the brain cells." Well said, Dr. Seuss. Our learning goals for this lecture are students to be able to understand the structures in fungi, animal, and plant cells, and also be able to understand the movement of fluids through cells. What we already knew is that protists are eukaryotes, just like animal cells, just like plant cells, just like fungi, and that eukaryotes are larger cells than prokaryotes, and they require organization for that reason. In the lab, we looked at many different kinds of organelles. We know that organelles are organized and membrane-bound, and they have a specific function that they have to play. Here's a image of our fungi cell. You probably want to pause and look at the different pieces here. This is a crude drawing. But what we're seeing is the cell membrane that goes around, nucleus that contains the DNA, vacuole for storage. In this case, see some phosphate granules in here. Bud scar here is a little bit of damage from the way that they reproduce. Mitochondria for energy, and cell wall. That's my animal cell. We see some of the same pieces. This is a little bit more technical drawing. We see some mitochondria right here, just like we saw. We see the here's the nucleus where my genetic material is maintained. We have my ER. This is the rough ER because there's the ribosomes on it. ER moves things around. The ribosomes are responsible for making proteins. We have my centrioles here for replication. My smooth ER here, some glycosomes, the Golgi apparatus, which is doing endo and exocytosis, getting things in and out of the cell, peroxisomes for cleaning up, all these tubules and filaments that are giving me structure across the cell to move. There's a lot of things going on here in the animal cell. In my plant cell, I see a lot of things going on too. There's the same Golgi pieces, the peroxisome cleaning up, mitochondria. We notice my vacuole here is very large. It gives us some kind of water pressure. It kind of holds this thing up, stores a lot of water here. Same nucleus, we get those filaments and the cytoskeleton moving around, the smooth ER, the ribosomes. We have some chloroplast in here. That's new. So here's our first half of our list of different organelles that we can see inside of these different things. The nucleus function is stores genetic information. The ribosome is where we're going to make the proteins. The endomembrane system, so we have the rough endoplasmic reticulum, and that's where the ribosomes attach at. So proteins are made at the ribosome and then processed and moved to the rough ER. The Golgi apparatus helps things come in and out of the cell and process those proteins. The smooth ER helping out with some fats. The lysosomes help out with digestion, recycling, breaking things apart. Peroxisomes also break some things apart. Does a little more chemical stuff, some different parts than the lysosome does. The vacuole is storage, holds things. The mitochondria, ATP production means energy. It's a powerhouse of the cell. The chloroplast is where we are doing photosynthesis, absorbing light to make sugars, which we can use for energy. The cytoskeleton holds things up, but it also acts as tracks that things can move around in the cell. The plasma membrane, also known as a cell membrane, is selectively permeable, and it maintains, lets some things in, some things out. The cell wall is outside of the cell membrane, and it's for protection and structural support. A lot of different pieces here. So if we're looking at fungi versus plant cells, a lot of the same thing in common. Plants have chloroplasts because they're autotrophs. They do photosynthesis to make energy for themselves. Fungi do not have chloroplasts because they are heterotrophs. They actually feed on other things. They both have cell walls, but they're made out of different things. Plants have cell walls made out of cellulose, which is has a little bit more bend to it than fungi does. Fungi cell walls are made out of chitin, which is a little bit more rigid. So same type of structure, but just a little bit different component. structure. If we're looking at an animal, a plant cell, and a bacteria there, we can see that there are some commonalities between them, but there are some differences as well. All right, let's do a developing question. Labeling. Pause and label. All right, let's see how you did. So we have the cell membrane right here. We can see it's pointing right here at the edge. We have this nucleus here. The nucleus is really easy to find because it's right up in the middle and it's always large and roundish. The nucleolus is inside, easy to find there. The endoplasmic reticulum is always surrounding the nucleus, so you're always going to find it right close. In this case, this picture didn't differentiate between rough and smooth ER. Here's my Golgi complex right here. It's always that kind of fatty, squishy part. We have 3 mitochondria in this picture here. There's lots of mitochondria inside of a animal cell. They only drew a few of them. This is the one they labeled. The lysosome, we're breaking things down. They didn't label a peroxisome. Vacuoles for storage. Vacuoles can be drawn in different ways, but they're always going to be kind of sac-like, kind of blimpy. And the cytoplasm is always going to be a hard one to tell what it's pointing at because it's pointing at, kind of nothing. So it's kind of a process of elimination. Be careful about that one. All right, surprise, another labeling. Pause, try your labels. Let's see how you did. So we have the same mitochondria. There they are right there. You can see they're a little bit different, but the drawing is still essentially the same. It's the same kind of mitochondria. The only difference between seeing in the last picture and this one's just from the artist that drew it. Round with kind of these little squiggly bits inside. There's my cytoplasm right there, again pointing at roughly nothing. Here they've labeled this is the rough ER. You can see they put the little dots on there for the ribosomes, and down here, here's the smooth ER. Kind of tendrils there, no ribosomes on it. Cell wall, you can see this is the cell wall for this cell, and then these other ones, this bottom line, these are cell walls for neighboring cells. The nucleus and the nucleolus, and here they've labeled the nuclear envelope, which surrounds the nucleus. Be careful, it's tough to tell which one's pointing at the nucleus, which one's pointing at the nuclear envelope, especially in this picture. This picture is particularly difficult. It's not real clear which one they're intending to label there. The chloroplast here, famously green, but they also always have these disc-like shapes inside of them. Helps you identify the chloroplast. The plasma membrane, you can see it's right inside the cell wall there. There's those peroxisomes right there. The Golgi apparatus, it's kind of bubbly. And here's a new one. This is the plasmodesmata. They're little openings, allow to move through the cell wall. All right, why don't you try to make a Venn diagram comparing plant cells and animal cells. Pause and draw that out. You should be able to get a few in each circle. All right, there are more answers, just these, just to give you some ideas. They both have mitochondria, both have Golgi apparatus, both have ER, both smooth and rough. They both have a nucleus, cytoplasm, ribosomes, among other things. All those organelles that are common to eukaryotes that that help them stay organized. Animal cells have no cell wall, where the plants do have a cell wall. Now be careful, they both have cell membrane. Very, very common to get confused. Some people will say, oh, well, animals have a cell membrane, but plants have a cell wall instead. No, animals have a cell membrane, plants have a cell wall and a cell membrane. They have vacuoles, but different kinds. Plants have chloroplasts, where animals don't. And, so some of the animal cells can have flagella. Like a sperm, for example, but not very common. All right, let's talk about movement across. We're largely talking about this because of the cell membrane, how things move around inside the cell. So diffusion is the easiest type of movement. It's just stuff spreads out. That's really all it means. If you've ever seen, some food dye dropped in water, it doesn't just stay there. It spreads out on its own. It doesn't take any energy. It just does it. If you have some perfume sprayed in a corner of the room, you don't have to turn on a fan to get that smell all across the room. It will just spread out. That's called diffusion. Now, whenever we have something like the cell membrane that's selectively permeable, it only selects for certain molecules. Some can move straight across it back and forth. Some, there's channels that can be open or closed by the cell. So diffusion can happen across the cell membrane depending on what it is, because the cell membrane is permeable to some things, meaning things like water. Water can just move straight across the membrane. It can go in, it can go out. Other things cannot, like glucose. Sugar can't come in and out freely. It doesn't flow across. It has to be moved across. Protein has to make that happen. So when we say selectively permeable, that's what we mean. Osmosis is similar to diffusion, but in diffusion, we saw the stuff spreading out in the water. In osmosis, we're seeing the water spread out. Because if we look at that dotted line there in the middle, that's a semi-permeable membrane. That means it's permeable to some things, in this case water, and whatever those little dots are, kind of salt maybe, they cannot move across. So what my water has done is, since the salt couldn't spread itself out, the water moved to try to dilute the, over here we see all this salt all bunched up and this salt all spread out. Some of the water moved over here to try to dilute that, to make it so that the water is equally spread out. The salt is equally spread out to equalize the concentrations. This requires no energy. Water will do that on its own. Natural process. We use 3 words to help us describe this situation. So hypotonic, isotonic, and hypertonic. Hypotonic means that there's less stuff, not enough. Isotonic means that there's the same amount. Hypertonic. So osmosis is finished when you end up with isotonic, the same same concentration on each side. Now we're talking about things moving in and out of the cell, going through the membrane. We have something called exocytosis and endocytosis, and we kind of get these little pieces that kind of get pinched off. See how they kind of, here's this thing, it kind of goes to the end, it kind of just opens up and releases. We can see how it's, step 1, it came from inside, it kind of pinched off this little bubble, kind of floated it across, and then when it gets it joins with the other parts, and there it's released. The exact opposite happens the other way. The only difference between exo and endo is direction. Exo means out, exit. Endo means indoors, come inside. All right, here's a developing question for you. Fill in the blanks with the names. All right, we got the nucleus, the ribosome, the ER, the mitochondria, chloroplast, Golgi, vacuole, lysosome. These are the big ones. All right, here's a harder one. Here we have organelles, location, talking about whether they're in plants and animal cells. We didn't include fungi in this list. Description and function. So fill in all the blanks. This should take you a little bit. Pause and spend a few minutes. Fill this whole thing out. Don't cheat. Honor system. Go on, don't wait for me to do it. Pause, go do it. You're only cheating yourself. Alright, you did it, didn't you? Here you go, take a look. There's the list. What's the difference between osmosis and diffusion? Well, they're both going to result in isotonic, evenly concentrated solutions. In diffusion, the stuff moves. In osmosis, the water moves. We usually see osmosis because we have some kind of a selectively permeable or semi-permeable membrane getting in our way.